Abarbanel from University of San Diego, from University of California at San Diego. He's a member both of the physics department and of the Scripps Oceanographic Institute, and he's going to talk to us about small networks of biological and electronic neurons. I can't read my handwriting, that's my problem. All right, well, Professor Abarbanel.
Okay. Can you push the standby button? It's on the, that's
right
it.
side.
Okay. In fact, while I'm talking, I can pass around an electron. Let's go back here. I'm going to talk about a very specific problem in neurobiology, but along the way, I'm going to describe techniques of nonlinear signal processing that are applicable to signals that, can we turn this down a little bit? Yeah, thanks. That are applicable not only to time series taken in biological samples, but in physical samples, in speech samples, anything that you want. So I'm going to talk around the specific biological problem, tell you how we're addressing it, why it's interesting, what we hope to learn, what we hope to accomplish, and I'll also emphasize in some detail the tools that we've used to address it. Okay, so this is where I am. I'm a physicist, I'm a theoretical physicist, and is there a pointer of some kind?
I think so.
Okay, and we have a group composed of some physicists, some serious experienced wet lab biologists, a few computer scientists, and some engineers. And we have been studying a biological control system in an invertebrate, in particular the California spiny lobster. This is a lobster that doesn't taste as good as the Maine lobsters. Makes a much better experimental animal because the measurements we make are made by putting an electrode through the cell membrane of the neurons that we're interested in. And the better tasting the lobster, the softer the cell membranes, and therefore it makes a very bad experimental animal. The California spiny lobster grows off the coast of California in the Pacific and south all the way to Chile. They're caught by local fishermen, and we simply purchase 5 or 7 a week. This is, and I'll show you a picture of the appropriate nervous system. The problem that we're interested in is the following. The lobster, which is a scavenger, comes out of its hiding place in the sand when it senses that there's some food to be scavenged. It stuffs it in a passive sack on its chest, like a shopping bag, and scurries back into its hole. After that, it swallows it, and in the stomach, there are a pair of clamps, mandibles, that clamp the food. And then there's a group of neurons, 16 of them, and I'll show you their circuit diagram, that govern the operation of a little scraper, which is a fork with 3 tines that simply scrapes the food and shreds it. That's its equivalent of chewing its food. When it's scraped and shredded, there is another group of neurons, which is the one that we study in more detail. There are 14 of them. Again, I'll show you the circuit diagram, that controls a pump or a swallowing mechanism, which is a little muscle chamber, and it sends patterned, nearly periodic signals to contract and dilate those muscles to pump the food that was shredded to the digestive system. We're interested in the properties of those 14 neurons that govern that pump, called the pyloric chamber. And this collection of neurons that acts as a control system is called a central pattern generator by biologists. I don't know what the central means, but the pattern is clear. It's the pattern of voltage outputs that causes this. We're interested in both how individual neurons in the circuit work and how they work collectively. And it turns out that the individuals, when isolated from the rest of the circuit, are chaotic. I'll tell you what that means. I'll tell you how we found that out. I'll tell you the implications of the analysis of that. When they are put into the circuit, connected with various kinds of couplings, which I'll describe, they act in a very regular way. So the regularization of chaotic processes to give functional output for biology is the goal of what we're studying. And I'm going to talk about that goal. I'm going to talk about the tools. I would call it the bottom line, but we're by no means done. The intermediate line is that we have developed, based on the analysis I'm going to tell you, a model, both a biologically inspired model and a phenomenologically inspired model. That phenomenologically inspired model consists of 4 ordinary differential equations, which I will show you. They are implemented on that circuit that's going around here somewhere. It's an analog circuit. The system itself runs at about 1 hertz, and so does the analog circuit. So it's not a fancy circuit, doesn't challenge Lucent Technologies' 10 gigabits per second transmissions, but that's how the lobster works, and our goal was to figure out how that works. I don't think that's part of, all right. Well, if you can read between the lines, as it were. We're really interested in design principles of this circuit. We would like to know.
Do we have a loose connector or what's going on? I've never seen phenomena.
We're fine here. Well,
We're trying to improve.
yes, well, the gradient is certainly up. All right, let me continue in words then while the technology catches up here. We're interested in, as I said, how the individual members of the circuit work, but we're also interested in the design principles. Because once we understand how this little circuit works, we have another goal, again, an intermediate line, not a bottom line, to take this biologically inspired control circuit and see if we can use it in a simple robot. We are nowhere near there yet, but I will show you how we are progressing along that path. Once we understand how this circuit works, we will feel somewhat confident about going to much more complex circuits. There are 14 neurons in the circuit I'm going to show you. There are 10 to the 12th in the circuit that's producing this talk. And there's the only things that are close about those are the logarithms, not the complexity. Okay, so here's more or less the outline of my talk. I've talked a little bit about the biological importance of the pyloric central pattern generator network. I will show you that network. I'll talk about some of the experiments we have done, how they are done. I'll talk about the nonlinear time series analysis, which may be the core of what you're primarily interested in. I would like to say, from my point of view, one can develop the nonlinear time series analysis in the abstract as a useful tool, make a software package out of it, which we've done, and you can use it. But unless you use it in the context of something which is an honest-to-God problem, whether it's a speech problem or a biology problem or a physics problem, I would argue that in some sense you're wasting your time and just generating numbers whose relevance may not in fact be known. In the case of the biological experiment, what we measure is the intracellular membrane voltage. That is the voltage between the inside and the outside of a nerve cell. It is this voltage through the generation of what are called action potentials, spikes in the voltage, that were described, well, for many years, but quantitatively first by Hodgkin and Huxley in the early 50s of the last century. It is through these voltages that the neurons communicate with each other. They communicate through either resistive couplings, which are generated by the two cells which are communicating with each other, literally abutting each other, and proteins go through the membranes and form channels through which ions flow with some resistance. So Ohm's law governs the connection between those neurons. There is a much more interesting connection in which a piece of protoplasm called an axon connects one nerve cell to the other. The electrical signal generated, and that's what we measure, as I said, propagates down that axon the way it would a waveguide. So low frequencies are strongly inhibited, high frequencies propagate very well. When the signal comes to the end of the axon, it releases what are called neurotransmitters that diffuse across a few hundred angstroms of the synaptic cleft between the sending neuron and the receiving neuron, and that chemical either stimulates or inhibits the action of the receiving neuron. So there are three different kinds of couplings: electrical, inhibitory, and excitatory. And in the network that I will show you, there will be electrical and inhibitory. In order to understand the network, you have to understand the dynamics of each of these elements, and I'll describe what we have done to do that. Fourier analysis on the signals that we measure are usually extremely uninteresting. It is possible to put the neurons into a periodic state or a quasi-periodic state in which the Fourier spectrum is either a sharp line or a series of sharp lines which can either be beat frequencies among each other, few incommensurate frequencies. But in the natural state, that isn't how they operate. So Fourier analysis is extremely unrevealing about the properties. And what we're going to do is to reconstruct the state space of the system from the measurements of a single voltage. And I'll describe the idea behind that and the things that we actually do. I'll talk about how we choose an appropriate time delay for the various components of the state vector that we construct using average mutual information. I'll describe how we determine how many components we need, which is equivalent to the number of operational degrees of freedom in the actual signal. And I'll describe a method that we call false nearest neighbors. These two little bits of technology, if you like, or software or algorithms, are completely general, completely applicable to any signal. They do not depend upon your having measured something from a neuron. I'll tell you how to choose the actual dynamical dimension of your signal. I will talk about constructing a biological model that incorporates a lot of the biophysics and biochemistry. And then I'll turn to a physicist's version of that model, which takes into account the analysis that we will have performed, the nonlinear analysis. I'll talk about the circuits, and then I'll finally show you the results of an experiment in which we actually went to the circuit, killed one of the living neurons, and replaced it with the guy that's coming around the room, and restored the biological function. So that at that point, we were convinced that not only did the physicists think that what you have in your hand is a neuron, but the biology did too, and that's sort of the final proof of the pudding. Well, I'll pass on that. All right, here's the beast. Well, it's the part of the beast that we take out. These experiments are not done on live lobsters. Once you crack the carapace, the shell, in order to get to the neurons, the lobsters had a very bad day. So we take the neurons that we're interested in an invertebrate, there's no backbone, there are ganglia, or a collection of neurons. We extract from the lobster what is called the stomatogastric ganglion that governs the digestive system and the chewing system. The lobster does have a brain. The brain consists of about 10 to the 7th neurons. That's too complicated for us. We don't deal with that. Presumably, sensory signals that say food is over there, turn right, come in here, and sensory signals that say the food is all chewed up, start pumping into the digestive system, presumably comes in here. We don't deal with that level of signaling or signal processing. We deal instead with what's called the gastric mill, which is the chewing mechanism, and we deal with the pyloric chamber here, and the muscles that are driven by the nerve impulses. Here are the two circuits that we deal with. This is the so-called gastric circuit. There are 16 neurons here, some of them repeated, as in this particular one that's called a gastric mill neuron. There are 4 of them. This gastric circuit runs at a nominal frequency of about 1/10 of a hertz. It is coupled weakly to the pyloric circuit, which is 14 neurons. This so-called pyloric dilator neuron is repeated twice, and these so-called pyloric neurons are repeated 8 times. What's not shown here are the nerve fibers that go from the PD, or the lateral pyloric neuron, or the anterior burster neuron, out to the muscles of the chamber. The muscles have in fact been cut during the preparation, but we can monitor the voltages that go down those nerve fibers to identify the operation of this circuit. And we can, in any given day, identify all of these neurons. We basically have a lookup table of the signals that they produce, and we measure a signal on the fiber that comes out of a neuron, and that tells you whether it's VD or PD or whoever. All of the connections between the neurons, I have a bigger picture of the pyloric circuit. All of these connections between neurons or among neurons are all well known. They've all been studied by the biology group that we work with, and over the last 25 years. This is the best known neural circuit around. There are about another dozen that are almost as well known. And this is really grist for the mill of a physicist, because now we really know the circuit, and the issues are, what is the design of the circuit? Why are there 8 neurons here? Why not 12? Why not 7? How come there are 2 repeated here? What is the nature of these connections? What is the strength of the coupling? This resistor is exactly what I described as a gap junction coupling. It is an ohmic connection between this neuron called the pyloric dilator and that neuron called the anterior burster. This and this are 2 of the chemical connections I described. When a signal voltage rises in here, it sends an electrical signal down here and depresses or inhibits the electrical activity that's going on here, and vice versa. So this is known as mutual inhibition, and these are 2 different kinds of inhibitors that use slightly different neurochemicals at the, over that transmitter. These diodes are meant to represent almost unidirectional, typically very weak connections between that neuron and this, or that neuron and that, and so forth. That's the circuit diagram. Every time you open up a lobster, one of these lobsters, that's what you get. This kind of circuit diagram, this central pattern generator, is quite typical of crustaceans and is found throughout animals, including in us, whenever there is rhythmic activity to be done. Chewing, swimming, walking, flying, we don't do that with these, and whatever. At the base of your spine, for example, there's a collection of about 30 neurons, another central pattern generator, that governs your ability to rhythmically walk or run, to move your legs like this. So what we have done at the outset was to have the biologist isolate individual neurons. So they can isolate this neuron, for example, by either physically or chemically cutting these connections. So they can turn off this connection, or they can physically cut the connection. And we can study the time series. And this is an example of the time series when the network is intact. We have not separated the neurons. So this is the firing of the AB and the PD, which are coupled in a resistive way, and they tend to fire together. Complicated, but together. The order 1 Hertz oscillation is this one. It's the envelope. These higher frequency oscillations, called spikes, are what are transmitted down the axons and form the basis of the communication from one neuron to another. The spike trains are the key in biological nerve systems. When you smell something or see something, the sensors, either in the epithelium or in the eye, send sequences of spikes back down nerve fibers, the optical fiber, or here to the olfactory bulb and onto the olfactory cortex, here down the optical nerve to the visual cortex. And the information about what has been sensed or what's going on or what's supposed to happen is all somehow encoded in these spike trains. In this particular circuit, that information is certainly in these spikes, but additionally, there's information in these lower frequency oscillations. The spikes have a broad spectrum ranging from 10, 15 hertz up to about 50 hertz. This is a 1 hertz oscillation. The LP neuron, which if you look back up there, it's a little bit inverted from the previous picture, is joined by inhibition to the PD. So when the PD goes on, it tends to turn off the LP. When the LP goes on, it tends to turn off the PD. This pattern, expressed in terms of signals down nerve fibers to muscles, is what, in fact, drives the functional unit, which is this pump. Up above, I'm repeating the signals in the intact circuit. This is what the signals look like when you isolate the neurons and measure their voltage activity. So this is voltage. The scale is over there. This is tens of millivolts. It's 15 in this case, to be precise. And this is the time scale. And you can see that while the AB retains a certain amount of regularity in its pattern, even though its signaling is quite different from that, the PD neuron or the LP neuron become completely irregular. And I'll show you a blow up of that. So what I'm now going to talk about, I think the data I'm going to show you is from the LP neuron. I'm going to talk about how we analyze that to determine how many degrees of freedom are in the activity of this signal. How many differential equations or individual terms in a dynamical system do we need to describe that? Doesn't tell us what the system is. It just gives us a clue, and that's the sort of the core of this talk. Here's a blow up of the voltage in some arbitrary units versus time. Our signals are typically sampled at 2 kilohertz to 10 kilohertz. 10 kilohertz is a fantastic overkill. 2 kilohertz is probably also somewhat of an overkill. We sampled them at 500 or 700 hertz. We'd capture all the oscillations at 50 hertz with no problem at all. One thing to notice about this, which I still find remarkable, is how incredibly clean these signals are. And that's, just technically speaking, is one of the great achievements that the neurophysiologists that we work with and the community of neurophysiologists accomplished over the last 25 years. The way you make the measurement is you take a hollow tube, little glass tube, you draw it out under a flame until the tip is about half a micron. You fill it with 3 molar calcium chloride as a conductor, and then under a microscope, you put that through the cell membrane. The tip resistance is tens of mega ohms, so even the thermal fluctuations might provide so much noise that you couldn't make these measurements. And so one of these experiments consists of a petri dish with the dissected nerve system, a microscope through which you look in order to put the electrodes in the cell, and a huge bank of filters that take out all the noise. And that's the technical achievement. This is the noise level. And those are beautifully clean signals. This is an example, not always found, but one that we always love to encounter, where we have no shortage of data, no shortage of clean data, and we can take data on any day on the same neuron and get reproducible results. So it's stationary, clean, and very low noise. Right? It's almost everything that you'd ever want in a signal, and probably from our point of view a very good place to start. That isn't true if you make EEG measurements. There what you're doing is putting electrodes on the skull. You're measuring through the skull. You're getting measurements which aren't well localized for any given neuron. This is a very fortuitous and good case in which to understand what the dynamics is. All right. Yeah. Oh, please. I'm sorry. Please ask questions. Okay. Speak up. Yes, sir.
Why do you attribute that lower portion of the signal to noise? Because there is inhibition, and on the other side you have also such a variation in the amplitude of the signal.
Well, the reason we do is that if you take out the filter bank, what you get is much more stuff here, similarly up there. The other is it's very temperature dependent. This is done at a carefully controlled, what you do is you take the nerve cells you're interested in, you put them in a saline bath so they live, and you keep that saline bath at 20 plus or minus half a degree. If you raise the temperature, you'll see these fluctuations go up. Also, if you isolate the cell by literally physically cutting it out of the collection of neurons, putting it in a separate dish, this will go down. So the general activity, background activity, also contributes to these fluctuations. That's why we think that's the noise level.
When you say that will go down, do you mean the fluctuations over there? Or
The fluctuations will go down.
But not the valley. The whole thing would go down.
You mean the bottom level of voltage activity? No, that's set by the concentrations of sodium, potassium, chlorine, and calcium inside and outside the cell by Nernst equation, which is basically a balance between the electrical forces due to different concentrations and the diffusive forces due to different concentrations. So this voltage and the top voltage there are typically in real units about minus 60 or 70 millivolts and plus 100 to 110 millivolts. And those are set not by us, but by the concentrations of ions. All right. Are there other questions? I'm going to now That's the biology. I hope you won't ask me any questions more about the biology. Okay. All right. So here we are. We have made extensive measurements of exactly a single scalar quantity of a system that is surely more complicated than a generator of of voltage alone. What's happening biophysically or physically in this? The nerve cell has holes in its membrane that are made by proteins that extend through the bilipid substance, which is the membrane, that allow ions, sodium, potassium, calcium, and chlorine are the main ions, to pass back and forth from the outside of the cell to the inside of the cell. So if you think about this from an ab initio point of view, you'd want to describe the current of sodium. You want to describe the current of potassium. You want to describe the diffusivities or conductances through these individual holes in the membrane. You'd want to describe a great many things. And in fact, the Hodgkin-Huxley equations, which one writes down for these cells, very often have a few times 10 ordinary differential equations describing all of these different currents and the rate of flow and the rate of opening of all of these holes in the membranes. I'm going to show you that this analysis on the membrane voltage, which is a coarse graining of all of those details, exhibits 4 degrees of freedom. And I'll try to tell you what we think they are, but that means that if you want to model this, you don't have to have 25 degrees of freedom. You only need 4. And I will show you the results of analyzing a model that we made that had 13 degrees of freedom that also exhibits 4 in the voltage activity. So this is phenomenological from which you can stand on a firm ground to make a model of what's going on here. Okay. So what we're doing is we're starting with a scalar measurement. Somehow or another, we have to reconstruct the possibility that the whole state space of the neuron involves more degrees of freedom. Now, what's in the back of our mind when we're doing that is either a differential equation that gives us the rate of change of this voltage, the second derivative of the voltage, however many derivatives we need in order to identify degrees of freedom, or equivalently, a vector made out of the voltages, a vector that specifies the state that in discrete time, the sampling time, in our case half a millisecond. Here, I'll stand up here so I can wave my hands. And we have a vector of quantities that describe the state that move along in discrete time. So you have a vector that tells you the concentration of calcium in the cell, but we're not measuring that. You have a vector that tells you the concentration of sodium, which we don't measure, tells you the voltage, which we do measure, and tells us how that evolves in time. This is a good time for any questions? Yes, sir. I can't see you very well, so you just have to yell, I guess.
So how do you know that there isn't, the cells aren't adapting when you take them out of the circuit? Maybe something happens when you take them out of the circuit and they realize they're out of the circuit and they change the behavior.
Well, you're right. We are making the assumption that this circuit is built from components which we can study individually. And the components are the individual neurons and the connections among them. You need some dynamics, not for a resistor, but you need dynamics for this process of the signal propagating along the axon, causing diffusion of a neurotransmitter across a few hundred angstrom. And we are absolutely making that assumption. The proof of the pudding is building the pudding and seeing whether or not we are able to reproduce the behavior not only of the individuals, but of the circuit as a whole. And I'll give you, as I said, finally, both results of an experiment that we've done that shows that once we built the individual, but also show you where we're going on the whole thing. Are there other questions? Oh, didn't get better yet. I guess we have time for more questions.
In your circuit diagram, you had a lot of inhibitory
In this particular circuit, there are no excitatory connections. And that tends to be true of these simple central pattern generators, not 100%.
Aren't there any excitatory connections from the outside coming in?
Yes. Actually, the way in which this circuit works is that it is turned on by neurochemicals that come from other parts of the nervous system. So the brain, to the extent that we understand what it does, sends a signal that says, please release neuromodulators. They then diffuse through the region in which these 14 neurons live, change the membrane properties a little bit, and cause them to start oscillating. And that's how we study them in that state. There are also some control signals from the muscles that go back to the neurons that say everything's doing fine, or they are not, change the pattern a little bit. And in the chewing circuit, there is a lot more control circuitry because the lobster has no way to know what the texture of the piece of food that it picked up might be. So if it's rough, this has to scrape harder. If it's smooth, it scrapes slower. All that control circuitry is not being studied here. That's really the next step if we want to make a whole lobster. But right now we're interested in, as I said, the designs of the circuit operating. Oh, yeah, it's.
Just unplug it.
Well, I think you have to be careful because you'll burn the light out if it doesn't have a certain cooling period. Okay.
Just put something on the back.
It's very sharp up there on the ceiling.
Right. Are there any questions about these formulas? How about that one?
What do we do?
Well, let's,
Are we missing something from the bottom?
at the top actually, but that's okay.
Well, the top it doesn't.
I'll just say
I
those
think
words.
we have to
Right.
resynchronize your PC, I guess. Yeah.
Oh, you want me to turn it off and turn it on again?
This is the synchronizing some odd resolution.
Oh, it thinks it's still broadcasting to that one.
Yeah.
Let's see how it goes, okay?
Okay.
I'll fill in. All right, so this is the data we have. And what we're going to do is record voltages at a lot of times. We typically make a recording at 2 kilohertz for a few minutes. So we'll have hundreds of thousands of data points. What we want to do is create a d-dimensional state space of vectors. The natural way to do that, the way you might immediately think of, is that dynamical systems, whether it's celestial motion or biology, is given in terms of various forms of Newton's laws, which involve derivatives of variables. So the first thing that you might think to do is to take this measurement and a measurement one time later, take the difference, divide by tau, and call that the derivative. Well, dividing by tau, the sampling time, doesn't do anything. But taking the difference between very accurate measurements is like a high-pass filter, and it emphasizes the noise, which you just spent 25 years trying to get rid of. So that's really not a very good thing. However, if you think about the new information that's in v at t plus tau minus v at t, you already know v of t. So v of t plus tau is the new information, and it was suggested some years ago that one should simply use that. V at one time and v, the voltage, at another time, is a nonlinear function of the derivative variables that you might expect to see. And there's a theorem that says that if you have enough independent samples of what's going on, and if the time is long enough, then the nonlinear system, in this case the neuron, will have evolved through the dynamics that underlie it to give you a new look at the system at an appropriate time later. What's an appropriate time? Well, in this system, everything's running along on timescales of milliseconds. It would be inappropriate to sample this circuit at a gigahertz, because nothing's happening in a nanosecond in this circuit. It's happening in 10 to the 6 nanoseconds. So you sample it at order of a millihertz. So what we want to do down here is to construct a vector made out of the measurement we made, the measurement, some integer, capital T is an integer, of the sampling time earlier. This is 2 times the sampling time earlier. That would have gone into the second derivative if we were going to construct it, and so forth and so forth. And I want to ask 2 questions. What is the appropriate integer number of sampling times, now that we know this is order of a millisecond, half a millisecond in our actual experiments, and how many components of this vector do we need? Those are the 2 things we need to get started here. All right. This simply repeats what I said, and I will go on to the next thing. All right. First thing that we do is we want the voltage at different sampling times, the components of this vector, to be relatively independent of one another. One way to discover their independence, or at least their linear independence, is to calculate their cross-correlation coefficient. It's actually autocorrelation because it's voltage correlated with voltage, and look for a 0 of the autocorrelation. Well, that in fact does tell you when they're linearly independent, but it doesn't tell you whether or not they are nonlinearly independent. And so what we do is something a little bit different. We turn to mutual information, which I'm going to think of, at least average mutual information, as a kind of nonlinear correlation function. What we do is from the observed data, we make a histogram of the frequency of occurrence of the voltage and the voltage at some later time. So we plot voltage along this axis, voltage at a later time along this axis, and just put a point in a box every time certain values occur. We also make the histogram of the appearance of the voltage itself. If you take the logarithm here to the base 2, so the answer is in bits, of the joint distribution divided by the individuals, and you average that with the probability distribution, that's the average mutual information over your sample, and it tells you the answer to the following question. On the average, over your sample, how many bits do you learn about the voltage at capital T samples later, having measured the voltage now, on the average? If you use this as a kind of correlation function, this quantity is semi-positive definite, so you're never going to, positive semi-definite, sorry, so you're never going to look for a 0. What you look for is a minimum of this, or at least that's a prescription. The theorem that underlies this says if you have infinitely accurate and infinite amount of data, it doesn't matter what time delay you use, but we don't have infinitely accurate, infinite amount of data. We have lots of it, but it's not infinitely accurate. So if you look at the first minimum of the mutual information, that is a prescription for choosing what time delay to use here. It's only a prescription, so if you choose to follow this method and you find that this time delay, in our case, it'll be 9 times half a millisecond, so 4.5 milliseconds, you should absolutely calculate everything else I talk about for 8 and 10 and 7 and 11 to make sure that they don't depend in detail on that choice. Why should you make a choice at all? Well, you don't have infinitely accurate data, so if t0 is chosen too small, it means that the system hasn't had a chance to develop enough to be able to tell the difference between the state dictated at time t and the state dictated sometime later. If you had infinitely accurate data, of course you could do that, but you don't. If you take the time too long, because these systems are chaotic and very sensitive to errors in the measurement, of which they always are, it means that the state you measure now, based on this voltage, and the state that you think you're constructing over here, measured based on that voltage, are completely uncorrelated in any way because of the nonlinearities in the system, which amplify errors exponentially rapidly. So this is a balance between those two things. In the case of our actual data, you can't see it very well. This is the average mutual information as a function of integers times the sampling time, which is half a millisecond. And I forgot how much data we had used there. It's just 32,000 samples, but you get the same thing. You can see that there's a very shallow minimum. And we chose that minimum at 9 times the sampling time, 4.5 milliseconds. And everything that I am going to tell you, we also check for 7, 8, 9, 10, 11, 12, because it's a prescription. It's not mathematics. The depth of this minimum, which is very shallow here, depends on the noise levels and depends on the quality of your data. If you did a computation using some of the models, you get a nice deep dip here. But this is real data. Yeah?
So they look for the first minimum. For the first.
Right. It's a prescription. Nobody says you can't take the second minimum or you can't take the second maximum, the first maximum being at 0 time delay, where, of course, your initial measurement is correlated with itself very highly. This is a prescription. The idea behind it is that the two components of your vector are rather independent looks at the dynamical system, but not completely independent. That would be 0 mutual information. If you go out very far, I've only gone to 20 here, you can calculate this as long as you want, the mutual information does tend towards 0 fairly rapidly.
One other question.
Yeah.
So, but in coming up with this measure of mutual information, aren't you making the assumption that the state of the system is almost completely specified by the voltage level? That's what it is. Probability of a given voltage.
Right.
It could be that the other properties in terms of how it actually changes, so this is question of second order properties.
You mean the concentrations of this and that, but the voltage, the great thing about this technique is that the voltage, or anything you measure that you can measure accurately, and you typically can't measure those other things as accurately in this case, but that whatever you measure about a nonlinear system, it and its time delays encompass all the dynamical variables of the system, although they may be a nonlinear combination of those variables you haven't measured. The voltage now and the voltage a little time later depend in detail on the concentration of calcium and sodium, on the amount of opening and closing of the various channels. But this is, as I said, a coarse-grained look at it in which all of that stuff is nonlinear and unknown in the way it enters here. And the theorem, which is a geometric theorem proved by mathematicians in the 1930s and sort of recooked up and fancied up for dynamical systems in the 1980s. The theorem says that if, in fact, you're dealing with a system of n degrees of freedom, where n is finite, And in principle, you could write down n ordinary differential equations that govern that, and you could look at the motion in an n-dimensional space. If you have 2n plus 1 of these variables, you can always unambiguously unfold the dynamics in the high-dimensional space that you have projected onto the voltage axis by your measurements. I can prove that theorem in a physicist sort of way for you, but I don't have, and if you want to see it, I'll tell you. The idea is really the following. You have a many-dimensional space, and the motion of the system through this state space is unambiguous. This is a specific calcium concentration, a specific sodium concentration, a specific voltage, and so forth. We're measuring everything with this structure projected down onto this axis. What we are doing by adding coordinates systematically, this one and this one and this one and this one, unfolding that projection, and looking for proxies, because this is a nonlinear combination of all of those variables. This is a different nonlinear combination because it happened 2 time steps earlier. We're looking for a space in which we have unfolded all of these projections. And that actually leads immediately into my next thing, which is how do we determine how many components we actually need here? The theorem...
I'm not sure what unfolding means here. I mean, are we trying to, you know, you said calcium and I don't know, something else.
Yeah,
something else.
other dynamical variables, right.
In other words, we're trying to find out, we're trying to resolve it into these.
No. No, this is a proxy space. The only measurement we have is voltage.
Yes.
All right? But I claim that if you measured this voltage, then the voltage this time earlier is dependent not only on that voltage, I'm going backwards in time, but never mind, but also on all those other dynamical variables, because I get it by integrating the unknown but actual differential equations that describe the system. So I measure the voltage now, and now I measure the voltage later. It comes from integrating this whole system of all those variables, so this is non-linearly dependent on all of that.
Yes.
So this stands in for all those things you don't know, and as long as these two are relatively independent by our previous prescription, it represents 2 independent looks at the system. If there are 10 independent variables, calcium concentrations and so forth, you need 10 of these variables in order to properly describe the state. That's what I'm saying. As for the, let me skip ahead to here. This is the picture in the state space that I'm getting to. What I've plotted here is the voltage 9 sampling times later, so 4.5 milliseconds, and the voltage 18 sampling times later. And this is a picture of what the state of the system looks like in this proxy space. Actually, we needed 4 dimensions, but this is close enough. This is the best I could do in a picture. And what's happening is those very slow oscillations are this. You can see the noise on it. And the rapid spiking is up here, and I'll describe what some of the dynamics are. When I talk about projection, the original measurements were taking these points, all of these points in this 3 or 4 dimensional space, projecting them onto this axis. So what we are doing by adding this axis and in our minds a 4th axis or 5th, is unfolding this projection. That's what I mean explicitly. Now, that actually gives us the clue to how to determine how many degrees of freedom are actually operating here, because what happens when you do the projection? Maybe I should start with this picture, right? Rewrite this talk. Start up here, that point and that point project down to here. And in fact, the bad thing that happens when you make the projection is that 2 points that in fact are very widely separated in the original space that you don't know get projected down to be very near each other, and instead of being true neighbors from dynamics, those are neighbors that follow along and then the trajectory folds back and comes in. They're false neighbors that get there simply by the geometry. So what we do is we go through our data set in dimension 1, and we look at every point and its nearest neighbor and ask, what do those points do in dimension 2? We know how to get dimension 2 because it's 2 voltages that we've already measured. If those points stay together, we call them true neighbors. If they move apart, the order of the size of this object, we call them false neighbors. Once we've done that, we go to dimension 2 and ask, look at the nearest neighbors in dimension 2, what happens to them in dimension 3, in dimension 4? When you're unfolded, you can't unfold anymore. And so the number of false neighbors as a function of dimension tells you what geometrical setting you need to describe the dynamics without ambiguity. That number up there is 40%. So what we have down here is dimension. This is the percentage of false neighbors as we go from dimension 1 to 2, dimension 2 to 3, and so forth. And you can see that when we get down to dimension 5, the number of false neighbors has gone to a small percentage. It's typically, that's probably about 1%. And at dimension 6, it's gone to 0. Once it goes to 0, remains 0. You've unfolded it. So this calculation, which turns out to be extremely easy to do, because in order to find nearest neighbors, you use, does everybody know what quicksort is? Anybody here read numerical recipes? Right? Quicksort is just a divide and conquer way of deciding who's greater than who. If you use quicksort in d dimensions, you can do this sorting on n points in n log n time, and therefore you can deal with hundreds of thousands of data points without drinking a lot of coffee. What this tells us is that whatever's going on in this neuron, as complex as you want to make it in terms of its microscopic processes, in the voltage activity, which is what we want to know for the communication among the neurons, you do not need more than 6 degrees of freedom, and I'll show you that 4 will do by going on to the next step of analysis. Now let me pause. This is really the critical signal processing point. Any questions? Has nothing to do with neurons, except you should use them. Yes?
Did choosing the sample information give much different estimates than the sample autocorrelation?
Sample, the mutual information? Not always, but I know some examples. 4 or 5 years ago, we used this method to study the degrees of freedom that are associated with the volume of the Great Salt Lake in Utah. Turns out it's measured every 2 weeks. It's now a tradition. I don't know why when the Mormons showed up there in 1847 they did this, but they measured the volume every 2 weeks since then. Okay? And I'm just giving you the physics of it and why it's interesting, and then I'll tell you the difference in the measurements. The Great Salt Lake has a watershed of about 100,000 square kilometers, so it averages over all the microscopic atmospheric fluctuations, and in some sense a good climatometer. Right? If you use average mutual information, you choose a time, I call it T0, of about 400 days, a little more than a year. If you use autocorrelation, you choose a time of 20 days. These variations are not monthly, they're annual and interannual, and that's the biggest difference I've ever seen in my life. Sometimes they're very close to one another. The reason I do it is because average mutual information is a property of the joint distribution function, and it's nonlinear. It has all the statistical information. It's easy to construct, and therefore I'm much more confident using it. That's really the reason. Other questions? Okay. Now, so we're already ahead of the biological game, which would have us using 20 or 30 degrees of freedom to describe every little biological process going on. But we can even do better. What I've told you is the global space in which this process, go back to the picture, evolves. So in this three-dimensional space, you can see that the slow oscillations come along here, they come up there, they go through some rapid spiking, come back down. Not quite periodic, but it's very close to being periodic, at least in this part of the oscillations. Just by eye, if we really needed a six-dimensional space to unfold this object, this would be a horrible mess, because we'd still be projecting from 6 to 3 dimensions. Here's how we find out what's going on. We go on to the orbits, which are called the attractor, because these are dissipative systems. We go onto the orbits, and we take a neighborhood of points that are unambiguously true neighbors, and we ask, how many degrees of freedom do we need in a local map, a local polynomial map, that takes us from this region of state space to this region? And we calculate that as a function of the number of neighbors we use in constructing this map, which we do simply by least squares, because we know all these points and we know all these points. Just make a polynomial map of order 2 or 3 or 4, whatever you like. We ask, when does it become independent of the number of neighbors, and when does it become independent of the local dimension? So that what we imagine, and it's often true, is that the local dynamics, of course, can be described by local polynomial processes, but the dissipation and the nonlinear dynamics takes this, twists it around, and you may need a higher dimensional space to unfold those twistings. A mathematical example that might appeal to you is if you have a dynamics that takes the plane and maps it into a plane, then it's clearly two-dimensional. However, if you make that dynamics live on a Mobius strip, then in order to unfold the Mobius strip, you have to have 3 dimensions because of the topology. We're looking for that two-dimensional local dynamics. If we do that, what you see here is these are different number of neighbors. That just tells us about the numerical fit that we make from local neighborhood to local neighborhood. And this is dimension of the local map. And you can see it becomes independent of dimension and independent of the number of neighbors when you get to 3 or 4 degrees of freedom. 6 is what we needed for the global unfolding. This tells us that the dynamical system underlying what we saw is 3 or 4 dimensional. It doesn't choose between these two, that's up to you. But it tells you really us, and this is really the critical point in the analysis of the individual neuron. We're nowhere near the circuit yet. It tells us that we only need 4 degrees of freedom, 4 differential equations to describe this. Let me pause again and see if there are any questions about that. We've unfolded things globally. We know the space that we have to work in order for the projection to be undone. And now we've gotten on to the motion of the system in that state space, and we've determined we only need 4 degrees of freedom in that space. This is clearly system very specific.
Let me just ask you, you have 4 degrees of freedom, but the variables, the intuitive variables, like you said, I don't know, potassium,
Right.
whatever, there are many more.
Indeed.
Yes, and that simply means that they are very correlated.
Yes, that's
And-
right.
And if you, I don't know, if you resolve it,
You can, in fact, biophysically go in and measure all those variables with different degrees of accuracy, but you can measure them. What this is saying is that after you have measured 4, you should be able, by modeling, to predict all of the rest.
yeah.
And we have, although we haven't done it for this neural system because it's rather complicated, we have done it for electrical circuits
But
that
they
we
would
built.
be some actual 4, right? I mean, the 4 are abstract. And
It could be, it could,
potassium, etc.,
yeah.
is potassium, etc.
right. But it tells you, no, it's abstract in the sense of voltages and their time delays. But if you actually go in and measure potassium concentration, calcium concentration, sodium current, and voltage, that's all you need. And now you want to know chlorine concentration, you can predict it on the basis of the dynamics of those 4. And as I said, we haven't done it for a neural system, but we've done it for a wide variety of electrical circuits. We
Does
figured,
that mean that those 4 that you mentioned first actually control the other one?
Yes.
Or does it mean that they...
no, it's what you said, that there's a dependence.
Yeah.
And mathematically what happens is the following. These are dissipative systems. So that if you start off in a multi-dimensional space, those 6 dimensions shrink due to the dissipation. And their orbits on the attractor, their orbits for a long time, live on a much lower dimensional space. And so there are automatically built in by the dynamics relations which are not always easy to discover among the variables you started with. And I think I have an example of that in terms of one of these Hodgkin-Huxley models, which also shows 4 degrees of freedom on the attractor, but starts off with a 13-dimensional space. And we know everything about the physics of the biophysics, I'm sorry, of the 13-dimensional space. Making the connection between the original space in which you can measure everything and this proxy space is not trivial. And I don't know how to do it in general. I'm sorry, yes?
Can you remind me again what the data that the basis of this analysis is? It's one of those neurons in the network?
Yeah,
Okay,
it's
and
the
so-
voltage of the LP neuron, which is at the bottom of the network, the way I draw it, isolated from the rest of the circuit, but still living, measured as a function of time. That's all that went into this. So that voltage I showed you, I can go back to it if you want.
Oh, no, it's
Okay.
I'm just wondering, does it change when it's in the network versus out of the network?
Yes, yes. No, and that was the question that was asked before. We are making the assumption, which we can only verify after we put the whole network together as a model, not as a lobster, that if we study the components, both the individual neurons and the nature of the connections, because the chemical connections have this dynamics of diffusion of the neurotransmitter across the synaptic cleft, if we model those correctly, we will get the intact circuit behavior from the components. And so far that's working out. It could have been wrong, right? All right. Now, there are other things that one can do, which I didn't put into this talk, but I will wave my hands about. What I'm going to describe is our attempts to now go on and make an actual model that works in 4 dimensions that reproduces the behaviors that we see. If I didn't know the neurobiology, even as well as I do, which isn't all that well, I might say, I'm not going to make such a model because I don't even know what to make the model out of. Here we know about various ions and various currents and so forth. I could do the following. If you give me an initial data set, or half of your data set, I could learn how each neighborhood in my appropriately unfolded space maps in 1 step of time or 2 steps of time into another neighborhood, and into another neighborhood and another neighborhood. And on the basis of that, I could make a predictive model. You give me a new voltage measurement. From it, I compute a new point in this state space, and I look at its nearest neighbor in the training set. I know where that neighbor went because I made a local model. I have a big lookup table of neighborhoods going to neighborhoods, and I would predict that that point that you just gave me would map into wherever its nearest neighbor did, and so forth and so forth. Those models can be made, assuming you have enough data, as accurate as the dynamics would possibly allow, because in a nonlinear system that has chaotic behavior, 2 points that are nearby eventually exponentially rapidly separate with a coefficient that's governed by the so-called Lyapunov exponent. We have made those black box-ish models of this neural behavior, which give prediction horizons as long as they possibly could be, because we also calculated the Lyapunov exponents. That was on the previous chart. So without knowledge of the dynamics, you can still make predictive models that are as accurate as they could possibly be given the dynamics. I didn't do that here. Here what we did is we turned to actually trying to make a dynamical model that first of all incorporated the right biology, and secondly incorporates this discovery of 4 degrees of freedom. So I'm going to move away from the time series analysis and into a little bit more of the biophysics. This is Kirchhoff's laws. You probably all learned it, whether or not you wanted to, as freshmen or sophomores in some physics course you were forced to take. It's conservation of charge. And it says the membrane capacitance, this is what a membrane cell looks like to a theoretical physicist. Membrane capacitance times the rate of change of voltage is the sum of the currents. What Hodgkin and Huxley did in the 50s, well, culminated in their work, but lots of other people contributed, was to write the currents, the sodium, J is sodium, there it is, potassium, chlorine, whatever, in terms of a certain opening probability of these holes in the membrane, a certain closing probability of these holes in the membrane, voltage minus the threshold, and a maximal conductance, which just tells you how much current could possibly flow through if everything were open. These are numbers that you can actually measure biophysically by techniques that were developed in the 70s and 80s. We made such a model. This model had 13 degrees of freedom. We had 2 sections of the neuron, so-called soma, which is the main body of the neuron, and a piece that related to the axon where the signals get propagated. And in addition, we had a detailed description of the intracellular calcium dynamics, which turns out biophysically to be really important. So we wrote these. I dare not even show you the equations of motion. They're horrible. They have lots of undetermined coefficients that have been approximately measured. But when you calculate the voltage from this complicated model, and you go through exactly the same process I just described, this is what the voltage looks like in its voltage time delayed, voltage 2 time delayed space. And if you go through all the calculations I just showed you, it is also has 4 degrees of freedom. So that tells you that 9 of those variables are predictable from 4 in the actual operation of the circuit, of the neuron, excuse me. What the relationship is, this doesn't tell you, and at this point we don't actually know. We're trying to find out, but it's complicated. All right, so here's what we did. I must say, to the horror of the biologists, although I think everybody in this room, are there any biologists in the room that I'm insulting? No, then I'll continue it. In a natural way that a physicist or an engineer might, we've learned that there are 4 degrees of freedom. So what we did is we wrote down 4 degrees of freedom and wrote down differential equations for it. Membrane voltage is clear. There's a fast current, which turns out to be a sodium current. We know that's there. There's a slow current, which turns out to be a potassium current. And it turns out from our study of the Hodgkin-Huxley model, we know that the calcium dynamics are important. Could you have written down 4 other differential equations? Probably, but these have a chance to be connected with the biology. So we were pleased with that. Now, how do we represent the right-hand sides here? In the Hodgkin-Huxley model, there's a standard lore of how to do it. In fact, you don't even have to think. You can buy software packages in which you just throw in all the stuff. We did something else following some British physiologists. We represented these right-hand sides by polynomials in the dynamical variables, by fitting the behavior of the system over the dynamical range of the system. And the actual differential equations are here, where these coefficients, A, B, C, D, and so forth, are determined by looking at the voltage current characteristics of the actual system approximately, and then of course we do what we all do, we all fooled around. The only thing that we know for sure is that these coefficients, which set time scales, they just multiply the time in the differential equation. This should be a slow current, so this is a number that's small compared to 1. And we know that the calcium variations occur on an even slower time scale, even though they're important. So this number is smaller than that one, smaller than 1. This was the model. Here's an example of the output of the model as a function of the current that we inject into the model cell. Let me explain what that means. If you take a real neuron and you inject very negative current, it takes those voltage oscillations and depresses the possibility of ever getting an action potential and excitation. As you raise the current, what happens is you get into a region in which the action potentials are permitted, and then when the current gets very positive relative to some natural current, the system simply goes into rapid action potential oscillations, and I'll tell you what that means dynamically in a minute. So at the top here is our model neuron, the one that was drifting around here, with large positive current being injected, and the current becomes more negative as you come down here, and if you keep on going, it simply turns off the neuron, which is exactly what it should do. And as you can see, over a wide range of currents, you get the kind of spiking, what it's called bursting behavior that you see in the actual neurons. Needless to say, we didn't know all of the parameters in our model. We chose them the best we could looking at the data from the actual neuron, and then we fooled around with them here. Now what's going on here dynamically, just as an aside, and is the origin of the chaotic behavior, is up there you can see that behavior is really periodic. That is not how the neurons behave unless you artificially put a very large positive current into them. If you do that, dynamically in that state space, you see a stable limit cycle, which is the periodic behavior. As you lower the current, this undergoes a sequence of bifurcations. That becomes an unstable periodic orbit, and you get unstable periodic orbits that are embedded in the chaotic behavior, and that's just nonlinear dynamics jargon for how you characterize the strange attractor. All right. Having made a numerical model, we decided that we would actually try to build it electronically and try to interface the electronic version, which is, again, somewhere around here, with the biological system to see whether or not we could make them work together. That was sort of our ultimate test. So this is the circuitry, plus a little bit of flying up there at the top, which is actually implemented on that PC board that I passed around. We did it by doing it on a breadboard. Once we got the circuits done, we ordered the PC board over the net. They cost about $10. All the wiring's done on the back. We populate them with parts that we buy at Radio Shack. They are resistors, capacitors, and some op-amps to do the multiplication, and they are an implementation of these differential equations. In addition, we have to represent the synaptic connections, where the voltage comes along, stimulates the diffusion of some neurotransmitter that diffuses 300 angstroms and causes the receiving cell to be inhibited or excited. We do that with a single differential equation, which is the concentration as a function of time, and there's a single time constant in that. So we solve this differential equation, and that's very simple, and here's the circuitry. We built that as well. All right. Now what we did, this is the circuit again, is we went into the circuit and killed this neuron. And the way you do that, of course biologists have a fancy name, you inject the actual cell with a light-sensitive dye, and you shine a light on it, and sure enough it cooks, and it dies. That's called photoinactivation. All right? And then what we did is we took the electronic neuron in a box where we had a little better control over all the resistors and so forth, and we hooked it back into the circuit and monitored the circuit by measuring the voltage here. And what I'm going to show you is the behavior before we kill the neuron, AB neuron, and the behavior afterwards when we hook in the electronic neuron. Well, that's the actual box. It is not a good idea to do experiments by trying to reproduce the parameters of the circuit with a screwdriver getting the angles right on those variable resistors. So we have a fancy thing. Inside this box, I think that's the next picture. Yeah. Inside this box is the circuit that I'm passing around, and then some interface stuff to connect with the neurons. All right. This is the intact biological circuit. This is the AB neuron firing by itself, that picture you've seen before. These are the voltages going down the nerve fibers that you monitor, and these are the patterns that the muscles have to receive in order to perform their function of pumping. We now kill the AB neuron, and these are the voltages down the nerve fibers, and you can see that there's no pattern here. It's almost as if, it's not a great analog, you've set these muscles in the pyloric chamber into an epileptic fit. It's going like this. And this is what's going on in the neuron that's still there, still alive, that we're monitoring. It's undergoing an extremely irregular firing. If you take this data set and analyze it, you find that it's a very high-dimensional, very complex oscillation. And here's the interesting thing. Up at the top is a measurement of the current that's flowing between the electronic neuron and the biological neuron, the PD, that we've now connected bidirectionally. It's very important that the electronic neuron be able to influence the biological one and vice versa. If you connect the electronic neuron simply as an external driver to this, what I'm about to show you doesn't work. This is the electronic neuron. This is the picture of the biological neuron I just showed you in the previous picture, and this is the voltage going down one of the nerve fibers. We connect them here. You can see that within a cycle, within a second, the biological neuron goes into a patterned oscillation. The electronic neuron goes into a patterned oscillation because it's now part of the circuit, and the pattern that we saw 2 slides back is reproduced. And the next picture shows you a different state of the electronic neuron. We can control that state by dialing our knobs. You put that electronic neuron into a slightly more regular state. This is the same biological neuron. They couple together and reproduce the patterns that we saw before. That's where we're going. Not just one, but we're going to do the whole circuit. And I want to say something, a summary thing, and then I'll tell you what we're doing with all this, why we were interested in it. Start off with voltage measurements on the biological counterpart of this guy. From that, we deduced by the procedure I described that it has 4 degrees of freedom. We made a model which we tested extensively numerically. We then built this thing, which is going around, that we tested extensively, and then we hooked it into the biological circuit. We're now fairly confident that we can build the entire circuit. Here's a PD neuron, Here's the electronic circuit that represents the diffusion of the neurotransmitter across the synaptic cleft, and so forth. We're building it all. We have several goals to the extent that we can really convince ourselves that what's going on here represents what's going on biologically. We can start asking questions that are impossible to ask biologically. The circuit I showed you, I guess I'll go back to this diagram. I would like to know why there are 8 of these. Why is it that this circuit has 8 of these guys in order to produce the patterns that go out to the muscles? Biologically, I can ask why are there 8 or 7 or 6 or 5, but I can't ask why aren't there 9. Why didn't evolution come up with 9? Maybe it's working on it, I don't know. All right? However, in order to do that experiment biologically, you've got to go to another lobster, take out one of its PY neurons, and sew it in here in some sort of way. You can't do that. However, in the electronic circuit, to the extent we have confidence in it, we can always add a 9th neuron, no problem, just turn it on. You want 6, we can do that. We can change the strengths of all these connections. In other words, what we have here is a really nice analog design tool for studying the design principles of a circuit like this. And again, to the extent that we believe we're reproducing the biology, can actually learn something about the biological issues here. Again, why 2 and not 7? Why 2 and not 1? Why 2 and not 0? And we don't know those answers yet. The other thing that we're planning to do, I alluded to, we're going to build the entire thing electronically, and we're working with a group at the Sandia National Lab that makes robots to see whether or not this biological control system can in fact be used to control robotic behavior. Controls in robots are typically linear feedback systems that are often very good, but they're not as robust as the biological systems. They don't do as well in unexpected situations like complicated terrain. So we're hoping to be able to do that as well. And I think that's probably a good place to stop. Yeah, I can't see you very well.
The neurons that there are 2 or 8 of, how do they interact with each other? What's going on there?
Oh, you mean what's behind this little picture? Well, that's what this symbol is. The 2 PD neurons are connected electrically. And I guess I'm going to talk tomorrow morning, and I'm going to tell you about experiments that we've done on the way these neurons are synchronized by varying this resistance. And these guys are thought to be connected by unidirectional weak couplings. That's what the diode is supposed to represent. But all those, I have a chart that actually unfolds all of that, and it's beautiful to behold, but hard to understand. But all those connections are known just as well as the ones I displayed.
Question.
Yes, please.
So now the dimensionality of all the neurons in the system are roughly the same, the 4.
Well, we have made extensive measurements on this one, this one, this one, this one, less extensive on that one, and extensive measurements on one of these, not all 8 of them. And they all come out to have dimension 4 or 5,
So,
depending on the environment, frankly, depending on sometimes the lobster you take them from.
it looks as if then the way you've laid it out, I mean, really the voltage is dependent upon the potassium, the sodium, and the calcium carbonates.
Right.
I mean, what else would there be that actually control the voltage?
Oh, well, in the Hodgkin-Huxley model, there are microscopic variables that describe the way in which the proteins that go through the cell membrane open and close. That dynamics is important.
Right, but they
And those are all, pardon me?
But they do control the inward and the outward potassium and sodium carbonates.
Yes.
So they have a direct implication on what is current.
Those are
Right.
responsible for us to sort that out.
Yeah.
Right, and those properties of the number that are open are measurable by so-called patch clamp experiments. I haven't talked about the chlorine concentration. I haven't talked about potassium currents that are dependent on the sodium concentration. All of these things exist in the detailed microscopic cell biology level description of the neurons. And what we're learning here is that all of those variables in the actual electrical activity of the neuron, or at least of these neurons, is not important, or excuse me, determined by 4 variables. And we chose the 4 for just the reason that you mentioned. Those are the natural 4. We could have chosen the chlorine concentration, but frankly, we don't know anything about it. Yeah?
So is there a theorem that says if you have a network that has components whose dynamics are of any dimension, like 4,
Yes.
that the network dynamics is at least 4, you know? In other words, when you put everything together, what is the underlying dimensionality of the dynamics of the network?
I don't know how to predict that a priori, or simply by giving you the network connectivity. But let me tell you about something I do know.
Well, maybe we can answer that later.
No, that's fine. If I take 2 of these PD neurons, I'm going to show tomorrow detailed experiments we've done when we vary this coupling between them. In the natural state, and in a whole spectrum of states, these 2 things are synchronized. So that instead of really having 8 degrees of freedom here, it turns out that in any practical sense, you only have 4 degrees of freedom, because even though they're chaotic, they're synchronized. We do not know what happens in the whole network yet. We intend to learn that both biologically, and that obviously is a little more complicated than electronically, in which we make measurements simultaneously in many cells. If they are in fact synchronized, which turns out to be an extremely important property of neurons, that's how our brain probably works. Various regions synchronize in order to communicate with each other, in order to pass on visual information to a decision system and back to the muscles. If they synchronize, the whole circuit, instead of having 4 times 14 degrees of freedom plus a degree of freedom for each diffusion here, could in fact be relatively low dimensional. And we just don't know that yet. Right? That would actually be a very exciting prospect, that you could replace the whole control circuit by a circuit of 4 is being ambitious, but maybe 10. Okay. But smaller than whatever 4 times 14 is. It's at 56. Sorry.
Yeah, I'm just wondering about the voltage trace that you showed earlier in the talk.
I can-
It was clear that there was kind of a profile to the maximum of the voltage response. And is that an artifact due to the filtering?
Just a second. Let me go back to it, and you can.
I'm trying to understand if it's a threshold, if it's a real threshold neuron or it's one of these neurons that doesn't have a real threshold.
Let's take this guy.
I mean, that could be a real threshold.
All right,
that could
so
be a-
these are the oscillations of the neurons in the circuit. And what is can you pose your question?
So the spikes that you see riding on the membrane
Yes.
potential don't all reach the same voltage. Is that an artifact of the filtering, or is
No.
that real?
That's real. No, the filtering is very high frequency filtering, because the noise, this stuff is above. It's typically to get some of the 60 hertz stuff out of the wall out, and so the filtering is typically 50 hertz and above, a low pass filter. These are, as I said, 15 to 50 hertz, but could we have filtered out some of the amplitude there? Well, it persists when you filter at 100 hertz too, so that I don't think is the issue. Yeah, I'm sorry, I just can't see out there.
When you chose to model neuron using 4 fairly critical variables, why you do that? And once you know there are 4 states in the system, why not just use the general modeling technique, show 4 states in there, print it up? Why is it important to use slow time, fast time, and so on?
The black box sort of modeling that I described, in which you model just having observed where this neighborhood goes in 1 step of 2 steps of time, and therefore be able to predict where a new point goes, works perfectly adequately. But it doesn't in any way have a tie back to the basic biology. And so from the point of view of determining a coefficients in a model that may apply when the environment changes, or may apply when the temperature changes, or may apply when it's hooked up to other circuits, biological circuits, you need a model that has some biological reality. The most biologically realistic model are these Hodgkin-Huxley models, but they clearly have complexity beyond what we need. And so our present activity actually is to determine how, from measurements, we can determine more accurately the properties of these simpler models, to determine them, the variables, as a function of very long time scale processes, environmental changes, and thus build a biological system that's more realistic.
But
Yes?
I can argue that your models, your four differential equations, there's not much biological reality there. I don't see anything in the world that would do this high precision division and multiplication that you have in there. Multiplying operators and divide operators. So unless you come up with primitives that are capable of implementing implicitly those functions there.
Well, the biological reality, to the extent that it has any, and I can tell you that we argue with the biologists about this all the time, is in two ways. One is it does describe a voltage and currents. That it certainly does.
Yeah.
And the coefficients are determined as best we can by the actual IV characteristics.
Polynomial function.
But it's a polynomial function over the dynamical range of variation.
Yeah.
It does not have the reality that the Hodgkin-Huxley model does in terms of diffusion coefficients of calcium. Okay?
Yeah, but I would be hard pressed to find in a neuron something that implements this division multiplication.
Oh, you won't. Absolutely.
So,
You won't. That's for sure.
I guess you can do that.
Mhm.
Oh, do you really need a model? Could you just use the data that you collect and say, given one point, predict the next one?
That's what you say.
No, you have to be able, you can do that, and that's what I described, but you have to do it in the right state space.
And then you would predict how the currents that you actually need vary.
No, not the currents. The only thing you can predict using this black box kind of modeling is the variable you already measured. Because you don't know the connection between calcium concentration and voltage until you make a model. All I know is voltages. So I can predict future voltages, but I can't predict calcium concentration because I have no idea what the relationship is until I make a model or make simultaneous measurements. And then, of course, I can learn the connection and make that prediction. And in fact, we have done, I alluded to the electrical circuits, not with the biology. We have studied circuits in which, say, you have 3 degrees or 10 degrees of freedom in the circuit, but only 3 active degrees of freedom. And we have explicitly shown that by measuring 2 voltages and then reconstructing a 3-dimensional state space from one of the voltages, we can predict what the second voltage will do.
I guess what I'm asking is how important is the role of the analog circuitry? If you had access to the measurements of all the things you needed, the state space and the variables that they're supposed to control, could you replace this whole thing by a computer with appropriate analog digital converters, digital and analog converters?
Let me answer the nearest question I know the answer to and then come back to yours, okay? We seriously considered implementing the differential equations on a DSP board and having an A to D converter from the biology to it doing the calculation and then a D to A converter back to it. But since these things cost $10 and that costs several hundreds of dollars plus lots of programming time, and they're a little bit too accurate to be biologically realistic. These things that I passed around are 5% and 7% parts. We didn't think that really got to the issue of reproducing the biology. It reproduced a model. Now, could we have taken data and basically had a big lookup table? I think we probably could have, if we could have acquired all that data and with enough accuracy. Turns out that it's really hard to measure, as I said earlier, the concentrations. It's really hard to measure the opening and closing rates of those proteins that penetrate the membrane without doing things that really change the cell. You have to suck up a little piece of the membrane, measure what's going on in that half micron of the tip of the electrode. And the simplest thing to measure very accurately is just the voltage. So probably one could do that. We haven't thought about it. Yeah.
So, for most of the topics been acting like noise has been an issue here, and I just like to get more of a feel for how noise interacts with the chaotic
Sure.
We go later.
We have a minute.
Go ahead.
Well, after that, the food gets cold.
Let's look at this particular algorithm, the false neighbors algorithm. Suppose you took a beautiful, clean system that you solved with 64-bit accuracy. You would find that the data from that, if it had 4 degrees of freedom, would come down here, slam down to 0 at 4, and just stay there. Now, if you add noise to that signal, what you're doing is you're adding a very high-dimensional system that whatever the dimension of the state space will busily occupy all of those dimensions, because although noise is not infinite dimensional when we really deal with it, it's extremely high dimensional. Maybe it's 10 to the 20 dimensional. So you would have to go out here a very long way to get rid of it. What happens as you add that noise is that this whole set of points rises, and as the signal-to-noise ratio gets smaller and smaller, it gets worse and worse. And if you take your favorite random number generator that you really believe in and start with signal-to-noise of minus infinity dB, namely only noise, you'll see that the false neighbor stays up there at 90 to 100%, depending on your calculational accuracy. So between here and there, you lose the ability to determine this. This turns out to be relatively robust against noise. And if you add 5 or 10% noise here, which is far beyond the experimental level here, you'll just see this whole floor rise up. And so if you were to see this come down and just stay here at some noise floor, you could still say it was a noisy signal, but maybe had 4 or 5 dimensions. As I said, you have a much worse signal-to-noise ratio, it all goes to hell. The local false neighbors, in which you actually get on the attractor, is much more robust against noise and still works accurately at 10 and 20% noise levels, and that's experimental. I can't give you a theory for that.